Okay, everyone, let's take a look at Mendel's dihybrid cross, or one of his dihybrid crosses. So, what is a dihybrid cross? So that is a cross between two true breeding individuals that express contrasting traits for two characters. And the example I like to use is the cross, the dihybrid cross, in looking at the characters of seed color and seed shape. So let's look at yellow and round true breeding plants crossed to green and wrinkled true breeding plants. So this is going to be the P1 generation, and the genotypes for these guys will be 2 big G's here for the yellow and 2 big W's for the round, and 2 little G's for the green and 2 little W's for wrinkled. And these are homozygous genotypes because the plants are true breeding. Now, to get the phenotype and genotype of the offspring in the F1 generation, well, what we should do is look at the types of gametes that can be made by each of these parents up here. Now, there's only one type of gamete that can be made by the yellow and round plants, because they're homozygous for the seed color gene and the seed shape gene, and the gamete here has to inherit a single allele for each of these genes. Well, the plant only has a big G to donate to the gamete and a big W to donate to the gamete. So all of the gametes will have a big G and a big W. The same thing goes for this parent over here. All the gametes will have one little G and one little W. Now, during fertilization, these will come together and we'll get the progeny with the heterozygous genotype for both pairs of alleles. And these will be yellow and round with respect to phenotype, right? Because we already determined that the big G is yellow and big W is dominant for round. So the next question is, now the important part of the dihybrid cross is being able to calculate the genotypic and phenotypic ratios of the F2 generation. And how do we get the F2 generation? Well, the easiest thing to do is just take a yellow and round here from the F1 and cross that to another yellow and round from the F1, and well, that's how you'll get the progeny of the F2 generation. And so we want to use these genotypes to be able to predict the phenotypic and genotypic ratios of the offspring from this cross. Let me get a new piece of paper. So we have, again, yellow and round crossed to yellow and round. These are two F1 offspring crossed to each other. Genotypes, again, heterozygous for both pairs of alleles. And I'm going to go over two methods for calculating the genotypic and phenotypic ratios of the F2 generation. The first method is the Punnett Square. I won't go over the whole thing, but I just want to show you how to set it up. And then I will use the forked line method to calculate the genotypic and phenotypic ratios all the way through. So for the Punnett Square method, what I like to do is take a look at all the different genotypes that can be made by the yellow and round plants that are heterozygous for both pairs of alleles. So all the gametes, well, of all the gametes, half of them will get a big G, and of those that get a big G, half of those will get a big W, and the other half will get a little w. So we have a big G allele. Half the gametes are going to get a big G. Of those, half will get a big W, or half will get a little w. So we have two types of gametes already. So some of the gametes are going to get a little g. Of those that get a little g, some will get a big W, and some will get a little w. Now, because of Mendel's postulate on independent assortment, all of these gametes will be found in equal numbers. So if you have 1,000 gametes here, 250 should be of this type, 250 of that type, and so on and so forth. And that's independent assortment. So as we will see that is because of independent assortment, I should say. As we will see in, I think, chapter 5 in a week or two, if these genes are linked, we may not see equal numbers of the different types of gametes. For instance, if, for example, all of the gametes that had a big G allele also had a big W, that would be linkage. If all of the gametes had a little g that had a little g also had a little w, that would be an example of complete linkage. Now, Mendel didn't see complete linkage. Complete linkage is theoretically, I think, impossible in most cases. So, but he didn't see any form of linkage in his crosses, which is good, which is probably allowed him to come up with his 4 postulates, because that would have made things really confusing if some of his genes were linked. But all of them demonstrate independent assortment in his crosses, in his dihybrid crosses. So for this part of the course, we're assuming none of the genes are linked, and we're analyzing crosses in characters that undergo independent assortment. And because of independent assortment, all of these gametes are found in equal ratios. So same thing happens to this plant over here, right? It produces 4 types of gametes. They're all identical to the gametes from the other parent because these plants have identical genotypes. Now, to get the F2 phenotypic and genotypic ratio, well, we just need to combine these in every possible way. Now, you could do this by hand with paper and pencil, trying to keep track of everything, but the easiest thing to do is to use a Punnett Square. And with a dihybrid cross, the Punnett Square is going to have 4 rows and 4 columns. (16 cells). So what you do is put the genotypes of the gametes from one parent along the tops of the columns, and the genotypes for the other gametes along the beginning of the rows. So we have big G, big W, big G, little w, little g, big W, little g, little w. And then we can do the same thing over here. Big G, big W for these gametes from this parent up here. Big G, little w, little g, big W, little g, little w. And then we combine these, and what we get is something like this. Now we can fill out the whole table like this, and essentially what we're saying is that of all the offspring that are produced, let's say 1,000 offspring, we can see that there are 16 different ways to combine the gametes based on the number of cells here. 1/16 of them are going to have this genotype. 1/16, 2 big G's and 2 big W's. And we can predict the phenotype based on this genotype, right? So they have at least one dominant allele for each of the characters. So these would be yellow and round. We can go down here and see when we have these recessive gametes that only have recessive alleles combined in the zygote to make this baby pea plant here. What we have is the homozygous double homozygous recessive genotype. So 1/16 of the offspring will have that genotype. That will be green and wrinkled. Now, you can get a particular genotype in a number of ways. So these happen to be unique in the corners here of the Punnett Square. But let's take a look at, let's say, this one right here, little g, big W, with this one, big G, little w. We get double heterozygous here. We get the same thing when you combine this one with this one down here. Double heterozygous. Same thing here. Double heterozygous, and same thing here. So we have identical genotypes up along this side of the Punnett Square. So we have at least 4 out of 16, which equals 1 out of 4 double heterozygous genotypes. So you could go through the whole Punnett Square and figure out the genotypic and phenotypic ratio of the F2 generation, but it would take a long time. There's a much more efficient way to do this. And the much more efficient way is called the forked line method. Forked line method. Now, let's go back to the cross of the two F1s. So a yellow round offspring from the F1 cross to another yellow round offspring from the F1, and the genotypes in the F1 were heterozygous for both pairs of alleles. And so the forked line method requires you to consider each character separately first. So pretend you're doing 2 monohybrid crosses instead of 1 dihybrid cross. And so the monohybrid crosses, well, you know, we could memorize, maybe you've memorized already the genotypic and phenotypic ratio of a monohybrid cross where both parents are heterozygous, right? But if not, you can do a Punnett Square. So the gametes from this parent, the gamete genotypes would be big G, little g. The gamete genotypes from this parent, same thing, big G, little g. And so we just fill in the Punnett Square. And what we have is 1/4 of the progeny will have 2 big G's, 2/4 big G, little g, 1/4 2 little g's. And same thing over here. Now we can just change the letters, right? So I don't have to do the whole thing because it's essentially just changing G's for W's over here. So the genotypic ratio is going to be 1/4 2 big W, homozygous dominant for big W's there, 2/4 heterozygous, 1/4 homozygous recessive. Now we can also do the phenotypes. 3/4 of the progeny will be round because the 3/4 of the progeny have at least one big W allele. 1/4 will be wrinkled. Over here, 3/4 will be yellow. 1/4 will be green. Okay, so now we can use the phenotypic ratios here of 3:1 and the genotypic ratios of 1:1 with the forked line method to figure out what the genotypic ratio and phenotypic ratio of the F2 progeny will be from the dihybrid cross. So how do we do that? Well, okay, here was our genotypic ratio for the seed color gene alone. Now, 1/4 of the progeny are going to be homozygous dominant, 2/4 heterozygous, 1/4 homozygous recessive. So we take those numbers and we print them out nice and neat, just like this. Now remember, there's the same ratio, genotypic ratio for the seed shape character. 1/4, 2/4, 1/4. And what I'll do here is, well, how can I explain this? The best way to explain this is, of all the offspring from the cross, 1/4 of them will inherit 2 big G alleles. Of those that inherit the 2 big G alleles, 1/4 will also inherit 2 big W alleles. 2/4 will inherit 1 big W, 1 little w, and 1/4 will get 2 little w's. Same thing for this guy. Of all the offspring, 2/4 of the offspring will inherit a big G and a little g allele. Of those that inherit a big G allele and a little g allele, 1/4 inherit 2 big G's, 2/4 get a big G and a little g, 1/4 get 2 little g's. And then we just repeat the process. The same thing happens for all those offspring getting 2 little g's. 1/4 of them get 2 big G's. Oops, sorry. Oh, did I mess up? I did. These should be W's. If you were paying attention, you caught my mistake. So, congrats. I'm glad I caught it before I went much further. So, 1/4 will get 2 big W's. 2/4 will get a big W, little w, and 1/4 will get 2 little w's. Now we combine these with just a little, just by multiplying fractions. So 1/4, 2 big G's, times 1/4, 2 big W's, is 1/16, 2 big G's, 2 big W's. And maybe you remember that from just a couple minutes ago when we were doing the Punnett Square. Let me sharpen my pencil. It's getting kind of dull. Okay, so we multiply here now. 2 actually. 1/4 times 2/4 is 2/16. GG, big W, little w. 1/4 times 1/4 is 1/16, 2 big G's and 2 little w's. Okay, and you just need to fill the table in. Down here, we're going to see 2/16, 4/16, 2/16, and then 1/16, 2/16, 1/16. All of these are heterozygous for the seed color gene. All of these are homozygous recessive. And you know, I like this method because I like patterns, and there's a pattern, a couple different patterns in here, and one of them is this one. So 2 big W's, heterozygous, and then 2 little w's. You repeat that here, and you can just see that by looking over at the letters here. Okay, so this is the F2 genotypic ratio of Mendel's dihybrid cross. 1:2:1, 2:4:2, 1:2:1. And I really don't like the F2 genotypic ratio. I guess it's kind of neat to see those numbers, 1, 2, 1, 2, 4, 2, 1, 2, 1. But the important one that we're going to see a lot when we start talking about gene interaction and epistasis is the F2 phenotypic ratio. And that one's going to be 9 to 3 to 3 to 1. And let me show you, and we could figure that out just by looking at these and saying, oh, okay, this one, every one of these that has a big G and a big W allele has to be yellow for the big G and round for the big W. So yellow and round, yellow but not round, yellow and wrinkled. And you could figure out the phenotypes of all these, and then you could add up the fractions. But there's an easier way to get the phenotypic ratio. And that goes back to so this is where we started the forked line method. Here are the F1 parents. We separated the characters one at a time. We figured out the genotypic ratios and the phenotypic ratios. So 3/4 will be yellow, 1/4 green. 3/4 round, 1/4 wrinkled. So we use those numbers, fractions I should say, with the forked line method. So in the F2 generation, 3/4 of the progeny will be yellow, 1/4 will be green. Of those that are yellow, 3/4 will be round, 1/4 will be wrinkled, right? Because we already determined that when you consider each character separately, you get 3/4 will be yellow, 1/4 will be green. 3/4 will be round, 1/4 will be wrinkled. So now of the 3/4 that are yellow, 3/4 of those have to be round, 1/4 have to be wrinkled. Of the quarter that are green, 3/4 have to be round, 1/4 has to be wrinkled. And we just multiply these. 3/4 by 3/4 is 9/16 yellow and round, 3/16 yellow and wrinkled, 3/16 green and round, 1/16 green and wrinkled. Okay, so that 9 to 3 to 3 to 1 is Mendel's F2 phenotypic ratio of the dihybrid cross. And of the 9, what does that mean? 9 out of 16 are dominant for both traits. 1 out of 16 are recessive for both traits. 3 out of 16 dominant for one trait. I don't know what I was doing with that slash there, for one trait. Then the other 3 out of 16 dominant for the other trait. So right here, this 3 out of 16 is dominant for one trait. That means the other trait is recessive, and they just flip for the other 3 out of 16. We can see that here. 3 out of 16 yellow and wrinkled, 3 out of 16 green and round. Okay, so that is Mendel's dihybrid cross. I think if you have any questions, you can email me or come to office hours or ask them in class. I'm sure you guys have several ways you can get a hold of me if you have questions.